Hello, everyone. My name is Dylan Jones, and I'm going to talk about how latitudinal gradients and species diversity provides open niche opportunities for poleward range expansions. So the latitudinal diversity gradient is one of the most widely recognized patterns in biological systems, with the greatest species diversity usually occurring at lower latitudes and lower elevations. Additionally, these trends can hold true for species abundances as well, within a single species having its greatest abundance at lower latitudes and elevations within its single range. Several mechanisms have been proposed for why we see these trends, with climate being a leading factor in limiting species ranges. However, climate change is causing shift in global temperatures and seasonality, and one response to climate change is that species are going to track changing climates and shift their ranges poleward and to higher elevations. However, species in a single community are not likely to do this in concert with one another due to physiological and dispersal limitations. So given the latitudinal diversity gradient, we expect that range-shifting species are going to encounter less speciose communities at these poles. And if niches are similar along these ranges, then we expect that less diverse communities will provide open niche opportunities. And so thinking about a niche being a space in which a species occupies for available resources with the pressures of competition and predation. And we think that in these poleward ranges, that niche opportunities, open niche opportunities are occurring through available resources or a reduction in predation and competition. And in these instances, range expanding species may experience increased demographic success, which I'll refer to ecological release, which is just the increased performance of the expanded range species compared to its native range. And this can cause negative impacts on ecosystems through invasion success. So my study system looks at the area, which ranges from Northern California up to Vancouver Island, here in green as its distribution. And the co-occurring jumping gall wasp. Gall wasps are a group of specialized phytophagous insects that deposit their eggs in plant tissue, where a gall forms, where that larva develops within that gall, feeding on that plant tissue. So my focal species here is native from Northern California up to the Puget Sound of Washington. However, it recently expanded its range in the 1980s onto Vancouver Island. When we assess the occurrence of our gall former on oak trees in the native range, generally this is what we see, with a fairly healthy looking leaf and a handful of galls occurring. That's what these dots are right here. However, in the expanded range, we can find hundreds of galls on a single leaf, not only on single trees, but across the island and consecutive years. And this could have impacts on tree health, which could cascade all the way through the ecosystem, which is causing land managers to be concerned for this unique habitat. One way in which species can experience ecological release is through the reduction of competition. And in phytophagous insect communities, they rarely compete directly. They more commonly compete indirectly through mediated pathways. So one of these pathways is plant-mediated interactions, which is a gall former can deposit its eggs on plant tissue, with the potential of changing the quality of that plant tissue, subsequently impacting another species. Additionally, we can have enemy-mediated interactions, which would be shared enemies. A parasitoid that might attack one gall former could additionally attack another gall former. So what I think is happening in our native range, that we have greater species diversity in our gall formers, which leads to strong indirect competition through those mediated pathways, leading to biotic resistance, a reduction in the ability of a species to outbreak. However, in the expanded range, I predict that open niche opportunities through the reduction of species diversity leads to weak indirect competition and ultimately ecological release in the expanded range. So I asked the question, does the latitudinal diversity gradient create open niche opportunities that facilitate ecological release in our range expanding species? I'll need to assess this at two different scales, the latitudinal diversity gradient, which will be at the regional scale, and the ecological release at the local scale. So I asked these two sub-questions. Does synaptic diversity follow the latitudinal diversity gradient? And if that's so, do regional scale patterns allow for open niche opportunities that facilitate ecological release at the local scale? So what we did in 2019 was we systematically sampled trees across 6 regions. R1 can be found here in green, and we move up in latitude to R6 in red here at the northernmost range of our oak tree. Each region has 6 sites within it, with a total of 540 trees surveyed across 3 surveying periods. we found 25 synipid morphotypes that we put into 3 gall group types. Here in blue is our detachable leaf gall formers that are just gall formers that don't occur integrated into the leaf tissue. Our focal species is also found as a detachable gall former. Next, I have in purple our integral gall formers, and these occur on the leaves integrated into the plant tissue. And then lastly, I have in orange our stem gall formers, given the name they occur on the stem. So when assessing diversity at the regional scale, we're looking across latitude, our 6 regions, and then at the local scale, we look within region. Statistically, our sites within region are not different from one another, so we pull across all 3 of our sites for within region analyses. The first thing I'm going to do is assess for latitudinal trends. My first diversity metric I have here is community composition, which is looking at the presence and abundance of gall formers within sites and ranges. Here are our gall formers, color-coded as previously seen, with our focal species here in black. Regions are also color-coded, and this corresponds to where they fall on the latitude range of our oak tree. Dots are sites, and ellipses encompasses those regions. So generally what we see here is that as we move up in latitude from R1 to R4, we have changes in community composition with some general overlaps. However, when we look at our expanded range on Vancouver Island, we see a very distinct community composition with no overlap with the mainland communities. We believe this is in part due to 2 factors. One is we have an absence of the detachable gall former diversity on the island. And additionally, our focal species is outbreaking on the island, having its highest abundance on the island. So with this, we see that a significant trend with latitude for our community composition. Additionally, for synipid richness, as we move up in latitude, we see a decrease in richness with the fewest number of species occurring on the island. Finally, our gall abundance excluding our focal species has a negative relationship with latitude. However, again, for our focal species abundance, we see a positive relationship with latitude due to it outbreaking on the island. So now that we do see latitudinal trends for our synipid diversity, we want to ask, do these regional scale patterns allow for open niche opportunities that facilitate ecological release, specifically in these northern latitudes? So at the local scale within each of our 6 regions, I'll assess for changes in competition. So the first thing I did here was a species correlation network, which is a partial correlation analysis. Our correlation strengths are here that correspond to these colors. These are the lines that connect our dots here, which are the gall formers. If it's significant, it's a solid line, dashed or non-significant. Our gall formers are color-coded as previously seen, with our focal species being here in off-white. But recall that this is also a detachable gall former. So we believe that galls strongly associated on trees equals potential strong indirect competition through the opportunities of species co-occurring, leading to competition. Additionally, species in the same gall group might compete more strongly with one another due to shared niche spaces or shared parasitism. So again, I did this for all 6 of our regions, and we know that species diversity decreases as we move up in latitude. However, species associations also are reduced as we move up in latitude, with the greatest number of species association occurring in these southern regions and an absence of associations occurring in the expanded range here. Specifically, our focal species associates with numerous other detachable gall formers in the southern regions that are completely absent on the island. So now we have evidence that gall formers do associate with one another on trees, providing support for competition opportunities. So we looked at trees where gall formers co-occurred with one another to assess for competition, and we predict that abundances will have negative relationships with one another if competition is occurring. So given this graph here. So what we did is we took our focal species abundance and ran linear models across our 3 gall group types. And we found that for detachable and integral leaf gall formers, we see significant negative trends, which we believe indicate competition. However, for stem gall formers, we saw no relationship, which we believe leads to a lack of competition with our focal species and stem galls formers. So the take-home messages here for my research is the latitudinal diversity gradient does affect our snippet diversity at these regional species pools. So we did see that richness and our community composition do change as we move up in latitude, which provides smaller species pools at these higher latitudes, which provides open niche opportunities due to changes in these competitor species, which can lead to changes in those mediated interactions that I mentioned before. So in conclusion, species are likely to move poleward as climate change continues. Poles are less diverse, so those range expanding species are likely to encounter less diverse communities, which could lead to open niche opportunities and ultimately ecological release. So it's important for studies to see how often range shifting species are going to experience ecological release and how biotic and abiotic factors influences these open niche spaces. So with that, I would like to thank my advisor, Kirsten Pryor, my co-advisor, Tom Powell, for all their guidance throughout this project, the field and lab help that we got throughout this project, landowners for allowing us to survey on these lands, and lastly, funding through the National Geographic Society, NSF, and Binghamton University. And with that, I'll take any questions. Thank you.